Ben olsam kullanmazdım.
Neden peki?
Neden peki? Çünkü Tanrı tarafından belki farklı düşüncelere sahip insanları da yadırgamak istemiyorum ama doğanın belirli kurallarının çiğnenmesi ve artık duygusal bağın kopmasına neden olur.
Yani ben kullanmazdım.
Neden peki?
Niye Allah'ın yarattığı bir şeyi niye değiştireyim?
Eğer ki biz kendimizi çok değiştirmeye kalkarsa geleceğimiz çok bozulur.
Throughout history, humanity has observed nature and wondered about the limitless variety it holds and the boundless beauty it beholds. This is why we ask the inevitable question, how? The answers did not come easily. However, now we have a substantial amount of knowledge about the world of genetics. But one particular discovery on the topic changed the course of genetics, as now we have the opportunity to go beyond observing. Now we can shape it.
The solution came from a bacteria that causes the most harm to humanity, Streptococcus pyogenes. In defiance to viral infections, the bacteria have utilized a system called CRISPR and located the genetic information of the attacking virus and precisely destroyed it via an enzyme, endonuclease, called Cas9. For Emmanuel Charpentier, who currently works at the Max Planck Unit for the Science of Pathogens, this was a remarkable observation and discovery in which she published her work on the topic in 2011. In collaboration with Jennifer A. Doudna from the University of California, they have transformed this defensive system into a genetic scissor that can precisely cut a DNA section and alter the sequence. In 2020, they are awarded with Nobel Prize in Chemistry as they enabled the path of many scientists to overcome problems with the help of genetic engineering.
The working principle of CRISPR-Cas9 genetic scissors is that the scientists create a guide RNA that is complementary to the targeted gene in the soon-to-be modified DNA. And thus, it will generate a Cas9 guide RNA complex which will bind to the DNA segment and cleave the gene. Hence, precisely cut DNA can be shortened or a new gene can be added on. Thence, when the genome repairs itself, its code will be transformed.
As a careful observation transformed into scientific breakthrough, the CRISPR also came with a new set of responsibilities. Within ethical grounds, this powerful tool can benefit to the humanity and nature. Without its exploitation, it can pave the way for new cancer treatments and scientific discoveries. Genetic diseases can be eradicated and the yield of crops can be further maximized to sustain the needs of the world.
Mutlu günler sevgili izleyenler. Bugün sizlere çok güzel bir paket hazırladık. Bir kavanozu 100 TL olan bu CRISPR kiti şu dakikalarda ekran üzerindeki numaraları aradıktan sonra sizlere bir kavanoz fiyatına bir değil, 2 değil, 3 değil, 4 değil, 4,5 hiç değil, tamına 5 kavanozu sadece 100 TL'ye gönderiyoruz. Ve siz bu güzel CRISPR'lara sahip olmak için kesinlikle ve kesinlikle yoğun bir şekilde arıyorsunuz. Çünkü hem kendinize hem de sevdiklerinize bu CRISPR'ı alabiliyorsunuz. CRISPR kiti 128 noktada doğal bir şekilde tamamıyla organik enzimden biyokimyagerlerin vızır vızır çalışmış olduğu bir ortamda bir şekilde elde ediliyor. Evet. Ve bu CRISPR'ları sizlerin sofralarına, damarlarına kadar ulaştırıyoruz. Hiçbir şekilde tedirginliğiniz olmasın. Almış olduğumuz tüm CRISPR'ları analizden geçirerek tamamıyla Türk DNA kodeksine uygun olan CRISPR'ları sizler için ayıklayıp sizler için bu CRISPR'ları üretime alıyoruz ve biz sizlerin sofrasına gönderirken tüm aşamaları geçirmiş oluyoruz. CRISPR'larımızın üzerinde üretim izninden tutun, üretim tarihine kadar her şey yazılıdır. Ve kendimize özel bir kavanozumuzla biz sizlere gönderiyoruz. Siz bu CRISPR'ları gönül rahatlığıyla enjekte edebiliyorsunuz. Aynı zamanda 2022 yılında da 2 tane ödül aldığımızdan dolayı bu gönül rahatlığıyla sizin çocuklarınıza da bunu tavsiye ediyoruz.
And then came this CRISPR, and the CRISPR, I think it's, I zoom in here. So the CRISPR protein and RNA complex looks like this. So if you just look in a little bit in detail, here you see this double helix. This is the DNA molecule that's going to be cut. And then these squiggly things, this is the protein. And we call this protein the Cas9 protein. And that's the RNA molecule. So every CRISPR enzyme has this protein called Cas9, the RNA, which we call guide RNA, and then it binds to the DNA and it cuts. So it's much simpler to do this kind of breaking the DNA and then repairing the DNA using CRISPR, Cas9, compared to these older methods. So everybody now uses this, and I think
medicine we take could be more efficient if they could be personalized. And I was wondering about how CRISPR could be useful in order to personalize medicine.
Yes, so yeah, that's a great question. You have to, you have to understand that every disease is unique and has a different molecular mechanism. The cause is typically different mutations in different genes. And so to be able to use this, we need to know what is wrong with what genes are problematic. Once we know that, then we can go and try to repair the mutations or the deletions or the aberrations in the genes that are responsible for that specific disease. So it's really personalized therapy, and not every cancer will have the same mutations. So that's one problem. Every disease will have a different therapy. The second is, even if you know which gene to fix, which mutation to fix, you have to somehow take this protein RNA complex, which is this CRISPR-Cas9, and insert it into this living cancer cell, for example. Or if you're thinking of an immune system disease, then you have to take it and put it into the immune system cell. If you think of a neurological disease, then you have to target the cells. But if you have millions of cells in our brain, you have to, and the mutation is affecting most of those cells, you have to target every single cell. So this is a big problematic. You have to somehow deliver the so the delivery of the therapy is a big problem.
During the process of genome editing, what if some kind of accident happens? What will be the consequences of this accident?
Yeah, this is a very popular question of targets. I don't think it's that big a problem because there are ways of preventing it. It's a case-by-case situation, so you have to every case is different. If you want to make a mutation, you have to make sure that there's no mutation anywhere else. And the problem is sometimes some genes are similar to each other in terms of sequence. So if your CRISPR-Cas9 goes and recognizes your target sequence, it may be recognizing some other sequence that's very, very similar. So the trick is you use tools of bioinformatics. So since we know the whole genome sequence of the human genome, you can try to target only sequences that are very, very unique. There are no other similar sequence anywhere else in the genome. But it's really not, in our lab, when we make CRISPR-Cas9 mutations, we use computer-based bioinformatics tools to find sequences similar to our target sequence, and we sequence that part of the genome, and typically there's really, if you choose it correctly, there's no problem with off targets. But of course, if you want to do this as a therapy in a human, you have to be much more careful. It's true for all types of drugs. You can make a protein that may work as a drug in the lab, but to actually take that protein and inject it into a human patient it requires a lot of controls and a lot of you have to make it in a very sterile environment called a GMP facility. All other drugs also are very regulated, and that's why they're so expensive.
I can say how the mechanism behind enzymes is really effective in terms of cancer research or stem cell biology.
The advantages of trying to target a stem cell, if you could make genetic changes in a stem cell, then every cell that is generated from that stem cell will contain those cells, those mutations, sorry. So an embryonic stem cell is after fertilization happens, so when the sperm and the egg meet, then the embryo, the zygote starts to divide. So you get the 2-cell stage, then the 4-cell stage, 8-cell stage, it starts to grow. And then it comes to a stage called a blastula. Blastula, because it's big, it's like a sort of a balloon, and it's kind of hollow in the middle. But inside there is a region of a lot of cells called the inner cell mass, and those are embryonic stem cells. And you can make modifications in the genomes of those cells, and the rest of the embryo, when it develops into a new baby, will have those mutations. So that's an embryonic stem cell. So in the laboratory, for example, if you want to make a transgenic mouse, I mean, you can't do this in humans, but if you want to do it in an experimental animal model, you can do that. You can modify the genes in the inner cell mass, embryonic stem cells, and then the embryos that develop will have those mutations.
Like, how do you think this technology will develop in the future? Like, what are some new techniques you think will be for this genome editing?
As I showed you in the slides I showed, it evolved quite rapidly. I mean, for maybe 20 years ago, people started thinking about how this would work, and then my lab, we invested a lot of efforts trying to use these old techniques like the zinc finger nucleases and the talons. They're very elegant techniques, but they're very difficult compared to CRISPR-Cas9 because it's much easier to introduce this RNA and protein into the cell for some sort of technical details. But so nobody could really sort of envision or foresee that CRISPR-Cas9 technology would be present even maybe 10 years ago. But as soon as it was shown to work, then there was this explosion and everybody started using it. So this is how science works. One scientist makes a discovery or has an idea and demonstrates that this is sort of useful, and then everybody jumps on the bandwagon and everybody's sort of using the same technique. So we can't really even imagine what will happen in 5 years or 10 years time. But right now what happens is there are many people who are making modifications of the CRISPR-Cas9 technique. So you can instead of making cuts and making mutations in the DNA, you can put like fluorescent proteins on it, and then you can go and look on make the protein bind, this Cas9 protein bind to your gene, and then try to visualize it under a microscope. Or you can make it sort of change it so that it works as a transcription factor, so you can activate the gene or suppress a gene. There is a lot of new variations of CRISPR-Cas9 to detect the presence of DNA molecules. Instead of making changes in the DNA, to use it as a detection method. Like, for example, when you have, when you go to the hospital with if you suspect you have the COVID disease, then the doctor takes some blood sample or some spit sample, and then we use this technique called PCR. But you can try to use these enzymes to detect the presence of the virus DNA. So detection technologies are very popular right now. not only for humans but also in like foodborne diseases. If you know which sort of bacteria or fungus is contaminating some foodstuffs, you can try to detect the presence of the toxins or the presence of this disease-causing bacteria. So those are very sort of out of the cell kind of technologies, not only changing.
Yeteri kadar paramız olsaydı büyük ihtimalle kullanırdık. Çünkü bunlar büyük ihtimalle çok fazlaca maliyetli şeyler olacaktı. Bizim de o kadar paramız olmayacağına göre şu anda günümüz şartlarında hani kullanmak isterdik ama kullanamazdık. Genelde kullananları televizyondan izleyen insanlar olurdu.
Daha önce hiç düşünmedim ama
Yani ister kendi üzerinizde ister işte çocuklarınız için kullanılabilecek bir şey. Kimi
genetik bir hastalık varsa
Aynen.
mesela onu tedavi etmek için kullanılıyor
Evet.
büyük ihtimalle. Onun dışında başka fiziksel bir özellik için çok mantıklı gelmiyor bana.
Yetiştirmek yanlış mı?
Allah beyin vermiş.
Evet.
Beyin verdiği için de diyor ki bunu kullanın. O zaman ne olduğu zaman diyelim ki Allah korusun kanser olduğumuz zaman kendimizi bırakalım. Allah ne dediyse olur. Bu ızdırap çekilebilir mi? Böyle bir şey olsa onları engellemek için gerekeni kullanırdık tabii.
Anladım. Çok teşekkür ederim.